Hi, my name is Celeste Peterson, and I'm a fellow at Harvard University studying microbial biofilm communities. So I'm profiling the bacterial communities by looking at their 16S sequence and how the sequence changes over time and space. And to do that, I'm using DGGE. This is not a normal acrylamide gel. It needs to be kept at 60 degrees when it's run, and the buffer needs to be pumped continuously. So we're going to warm up the buffer for about an hour until it's at 60 degrees, and then we can run the gel. Now I'm going to wash this plate with dilute alkanox solution, feeling around for impurities and dried acrylamide and things like that to get rid of. Anything like that can cause irregularities in the gel. It's important to wipe off all the water. So when I assemble the gel, I will put that chip on the outside of the gel. So the gel will be in between these two pieces of glass, and the chip will be on the outside, and it won't affect the gel in between. So now I'm going to assemble the gel in these clamps. And then I squeeze them together and tighten by turning to the right. As with any gel, it's important to make sure that the bottom is flush to prevent leaking. We now turn the gel around and put it in here to get sealed with a gasket. So it's important when you do this that the plates are actually lined up with the gasket here. So these knobs on the side then go in, and you press down to tighten the seal. So this is a 30 ml syringe, and this part and this part come with a bio-red kit, and it just fits on like this. And that's just to get rid of the excess water in the tubes. To put the tubing onto the gel, to load the gel, first you put an adapter into the tubing, and then a needle into that small adapter, and put it right in the center of the gel, just so it's barely below the bottom glass. Then this Y adapter goes into the long tubing, and it's going to connect to both syringes containing the high and the low density solution. So these are pre-prepared solutions. The blue one, I've added a dye here that just indicates that this is the most denaturing solution, and this is the least denaturing solution, the low density one. And they have everything in them, and now I'm going to add Temmet to make them polymerize. Temmet is going to be added. This one I actually used. I have twice as much of the low density solution. And once you've added the Temmet, you have about 5 to 7 minutes to fill the gel before it polymerizes. Now we fill the syringes with the solution. So I'm going to fill it with about 15 mils of solution. And then to get rid of any air bubbles, I'll turn it upside down and squeeze gently until that top air bubble is gone. Now this little screw here is going to go into this groove. This adapter is going to line up with this clamp here, and it's going to get clamped in very tightly. And then this tubing is going to attach to the syringe. And the same thing is done on the other side with the high-density nitrate solution. Where it's time to deliver. And so I'm going to be turning this wheel in a steady clockwise motion. And first, the blue solution is going to go out first, because that's the most denaturing, and it's going to form the bottom part of the gel. And then after a little while, the clear solution will start filling in the gel, and a gradient will be formed.
One moment.
Okay. And the comb is inserted gently on an angle to avoid producing bubbles. Important as soon as you finish pouring the gel to wash out your tubes immediately, because they will polymerize. Down so this little notch is below the black. And on the other side I'll do the same thing. Set it on this rest thing. Hi, my name is Celeste Peterson, and I'm a fellow at Harvard University studying microbial biofilm communities. So I'm profiling the bacterial communities by looking at their 16S sequence and how the sequence changes over time and space. And to do that, I'm using DGGE. So DGGE is denaturing gel electrophoresis. And basically, the idea is that there's a gradient, and the DNA, as it runs down the gradient, gets denatured. And so different 16S species will stop at different points along the gradient. And so it ends up being a fingerprint of the different 16S species in the community. So the goal of my experiment is to see who is in the community and how that's changing over time and space. And I can run the 16S DGGE gel, and then I can cut out the bands and identify the species that are represented in each community. So one of the biggest issues when doing a DGGE gel is to figure out the gradient that's the best. And so that is just something that needs to be played around with and figuring out for a particular community which type of gradient. So the one I'm using today is 35 to 55% urea. One of the big problems that can happen with this technique is that smears come up in the lanes. And I found that for whatever reason, one of the best things to do is to run the gels for a really long time and then the smears disappear. Well, today I'm just using it for general bacterial communities, but this can also be used for a specific family of bacteria to see how those change over time. And those tend to be very clean gels. You can also look at mutations in single genes with DGGE.